My name is Nora Perrone-Bizzozero, and as Tom said, I'm a professor in the departments of neurosciences and psychiatry and behavioral health. And I've been at UNM for about 30 years, so I have been here for a while. Nonetheless, I wanted to tell you that I have been working on the genetics of schizophrenia and the biology of schizophrenia for a number of years, starting when I was in Argentina, where I am from, and in the University of Buenos Aires, where I did an internship in genetics and biochemistry. So I have also PhD in biochemistry, and then moving to Harvard Medical School at the Department of Psychiatry in 1984, where I did my postgraduate work for 6 years. I was there first as a postdoc and then as instructor in psychiatry until 1990 when I joined UNM. And you know, my background is both genetics, molecular studies of brain development, and I also have done, you know, neuropathology studies, and I'm also doing now, my focus is also on the molecular mechanisms of substance use. So, but coming back to the presentation, some of you may have attended and remember that I presented about 18 months ago a talk about the basic mechanisms and the genetics of schizophrenia, and for today I wanted to give you an update. So I'm going to share my screen now, and we'll start. And if you have questions, you can use the chat, and I will be happy to answer questions at the end. So again, thank you for the invitation to all of you to the NAMI Santa Fe. As I said, you know, my first presentation is just to tell you that schizophrenia is a complex polygenic disorder, meaning multiple genes with thousands of genes with small effects. So it was a hard disease to study the genetics of schizophrenia. And for now, for my update, I'm going to talk about genetics and epigenetics, and I'll tell you this is an emerging field that is very important in mental illness research. And also I'm going to talk about other major mental illnesses such as bipolar and the relationship with schizophrenia. So, okay. So quickly overview. So first I'm going to review this. Some of the material presented 18 months ago, but after COVID hit, it looks like it was 10 years ago. So I don't know how, but everything looks like it's far away. So, and then I'm going to tell you something new about the polygenic risk score and how can be used about diagnosis. What are the missing contributing factors, epigenetics? And finally, I'm going to leave an exciting field of neuroimaging genetics and some of the really approaches that I think is the future of diagnosis and treatment. So as you know, schizophrenia is a thought disorder affecting about 1% of the population worldwide, every country, every continent. Age of onset is late teens and early 20s, especially earlier for males. The cases, most of the cases are sporadic, but still highly heritable, and is diagnosed by presence of both positive and negative symptoms. The positive symptoms are the psychotic symptoms, and obviously they are diagnostic by the presence of delusion, hallucinations, disorganized thought, but also there are negative symptoms or deficit symptoms, and they are diagnostic by their absence. So I can, I'll give you copies of all my slides, and I put all these words starting with A. My accent, I still have a very heavy Spanish accent, so I'm not going to pronounce all of these words like apathy and hedonia. But the most important is all of this is lack. So lack of interest, lack of or inability to feel pressure, lack of ability to convey a clear speech, apparent lack. It's apparent because obviously this is what the physicians, psychiatrists are seeing, lack of will and lack of desire for social interactions. In addition, we have cognitive impairments in many of the patients. And what is important to appreciate is they are heterogeneous symptoms. So not all the patients present all the symptoms all the time. And some of the symptoms of schizophrenia may be present in other illnesses such as bipolar disorder. So it's kind of very hard to diagnose. And so again, there's no biological marker and everything is dependent on the clinical symptoms. Sorry, I get in here. So how we know schizophrenia is a genetic disorder? Well, this was the work done in the late 60s and early 70s by Erwin Gottesman and Seymour Kettin, and they were doing twin adoption studies. And basically, as I said, the risk of predisposition in the general population is 1%. And you increase, from the secondary degree relatives to the first degree relatives, the risk of predisposition increases. But when you reach identical twins, they have the same genetic, the same DNA sequence, then in that it's only 48%. So you can tell, students always ask me how you have 50% and you still have high heritability. We are going to talk about this later when I talk about epigenetics, but suffice to say that they are also in the discordant twins, so the twin that does not have schizophrenia. They may have some eccentric behaviors without being ill, and also they can pass the disease to the children. So they are not ill, but the children could have schizophrenia. So we'll go over this more later, but I wanted to tell you, so this is nature versus nurture. How we know what are the contributions, what factors weigh in. And in these twin studies, there have been a lot of studies in all these years. And this is from Patrick Sullivan. There's a meta-analysis and shows that the additive genetic risk of all of these studies is about 80%. And then obviously if the twins are raised together, there's going to be a shared environmental influence that is 11%. And you are seeing, obviously, if you add this together, it's 92%. So we have 8% that still is missing, and it can be explained by other factors as epigenetics. So, I'm going to give you a very, very brief introduction to human genetics because the data I'm going to present from original articles, I want you to be able to see the real data as they published, require a little bit of background. So the common sources of genetic variation. So you have eye color, hair color, we are all different, height, weight, everything is different. So the best, it can explain in 3 different ways. The single nucleotide polymorphisms, which is the focus of the studies, and I'm going to discuss in a minute, but there are other variable number tandem repeats, copy number variation. Those are the common. Rare variations, there are some chromosomal abnormalities, and you might have heard of the DiGeorge syndrome or 22q11, that is a deletion in this chromosome 22. And what happened is a very severe developmental disorder, and the risk for schizophrenia goes from 1% to 30%. So they are rare chromosomal abnormalities, but they are the minority. And then, so this is at the level of DNA, we have this. At the level of how the genes are expressed, we have epigenetic factors and methylation, which are the more methylation in the DNA, the less expressed is a gene, and histone modification like acetylations that do the opposite, increase gene expression, and there are also non-coding RNAs and microRNAs that we study in the lab. So again, what is the, the single nucleotide polymorphisms I call for brief SNPs, and I apologize, it's a long word to say all the time. So here is the double helix that you must be familiar with. And here you have bases. The A is adenine, thymidine, cytosine, and guanine. So these are the ATCG, and they're paired in this way. During DNA replication, you can have one mutation here. This E is now a T, and because T is paired with A, you have what is called a small mutation and one base. But remember, I didn't tell you, sorry, that there are multiple bases. It's 3 billion, so it's a large number, as you can see here. And there are about 50 to 100 bases in between the SNPs. So we have so much variation, up to 60 million SNPs, that make us different. And 70 of those could be new for each generation. So this all contribute to variation in the genome. But how we use this information is to link mutations to diseases. And I'll explain this with this diagram, and you can see here a chromosome from the mother. Actually, it's a chromatid, so half of a chromosome, and a chromosome from a father in blue. And what I wanted to tell you, these are the ancient or ancestral chromosomes. And let's assume that the A here, this here is a mutation, a SNP, that or other type of mutation. And then all the subjects with this A mutation have a disease, but those that do not have it do not. And after many of generations of recombination, and you can see all the chromosomes crossover, then you reach an equilibrium in which the A mutation is always in the same LD block that is inherited together. You cannot break down this. It's the minimum unit that you inherit. It's always close to a SNP2. And this is called tagging SNP because it's not the disease mutation, but it's very close to it. So if you have a chip that tells you, oh, the allele here is a T instead of a C, then you can say, oh, these subjects may have the mutation and may have the illness. Okay, so this has been done in higher scale in studies called genome-wide association studies, or GWAS for short. And here we have thousands of patients and thousands of non-patients that they are not related. In this case, it's just from the population. And we isolate the chromosomes and the patient DNA, and we probe this gene chip, they are this size. And then we probe with the non-patient DNA another chip. And then we compare what is different between this and this. And then you can see how many SNPs are different. And with that information, you can relate which of the SNPs are associated with the illness that these patients have. Okay, and then we'll talk about how you do this statistically. But the main, for all mental illnesses, the main consortium that does all of these GWAS studies is the Psychiatric Genomics Consortium, and it's the largest consortium. It started in 2007. There are a lot of scientists, 38 countries, more than 1 million subjects, not all in the same, from the same disorder, and I'll tell you how many disorders they are studying now. But it's important to say that for schizophrenia now, they have more than 100,000 patients, okay? So there is a website, it's called GWAS Atlas, in where you can ask how many GWAS studies are for each disorder. So here I enter, this is the website, I enter, okay, I want to know of these, all the GWAS that have been done in schizophrenia, and so far there are 6. I have them all through the year and how many patients. I said about 100,000. And they have these publications, of which I'm going to tell you about this and about this. So 3 publications, the last 3, I'm going to tell you today in a minute. So the first one I think is the most influential for schizophrenia. It was published in the prestigious journal Nature, and is the biological insights for 100 schizophrenia-associated genetic loci. Loci is a region in the DNA, and they had about 37 cases of patients and more than 100,000 controls. And in this, they found 72 of the 108 independent loci. So they don't segregate together, so they are independent. And they have 72 that are associated with protein coding genes that are related to schizophrenia. And one of the ones, the most significant one, is the dopamine receptor 2. And you may or may not know, but this is the main receptor that is targeted, inhibited by antipsychotic medication, all of the antipsychotics. There are other receptors that are associated. And there was another, like glutamate is another neurotransmitter that has other type of receptors. And Eli Lilly had a drug for this metabotropic glutamate receptor 3 that didn't pan out, but they are pursuing other glutamate drugs, and then calcium channels that we'll talk more later. And I want to emphasize one of the benefits of linking genes to diseases is not only that it can give us more effective diagnosis tools, but it can give us better targets for drug treatment, something that we call personalized medicine. So it's the best treatment for the patient. And you have heard probably in cancer, there are personalized medicines depending on the genetics of the tumor, so you can target a drug to the tumor. But in psychiatry, you can do this as well. And they have some, I give you this example, a review that is very good in terms of all the studies about drug medications and genetics, what are the best. And also a recent study that they analyzed using genetic analysis to look at which patients will benefit from clozapine. So this is, again, it's a developing field, but it's very promising. So coming back to the 108 risk loci. So let me introduce what is called a Manhattan plot. And the idea of the Manhattan plot is like, if you see like this, think about Manhattan and the skyline and all the buildings. And the highest building means the stronger the finding. And these are all the chromosomes here and the X. So we have the 22 autosomes and the X. And basically what I wanted to tell you, for example, that was something that we didn't know when this was published in 2014, is that the major, the tallest building had to do with the immune system. And now we know the immune system plays an important role in the development of schizophrenia, mainly by prenatal infections that I'm going to tell you, but this something was surprising and is something that we are not using right now, but I'm hoping that this is going to lead to new medications in the future. And then a couple of other variants. One is a microRNA, it's MIR137 that I studied in the lab. And one is a calcium channel that I highlight here because it's also important for bipolar disorder. So this is chromosome 1. I'm gonna tell you more about MIR495, and this is the work that Carrie Wright did in my lab when she was a grad student. And my collaborators for this work is Jessica Turner and Vince Cancun, where they were at MRN and close to UNM, and now they are still collaborators. They moved to Georgia State University. So here I'll give you an example. We have like 6 or 7 papers on this, but the one I wanted to tell you about is that if you look at polymorphisms in this gene and regulated genes, and then look at the brain structure of the patients, what we found is that the risk alleles here, I don't know if you can see it, the TT allele is a risk allele. And the more of the risk allele and other risk-related genes the patients had, the less thinner was the cortex. And cortex is important. So the cortical thinning is one aspect that schizophrenia neuroimage has found all over. So we can now link maybe some of this to a particular variant in this gene. But the PGC has more than schizophrenia. And you can see they have Alzheimer's, anxiety, OCD, substance use. And what I want to tell you now is the cross disorder group, in which they try to see the risk variants for schizophrenia, autism, ADHD, bipolar, and major depressive disorder, and trying to see if there are common variants that they are present in several of these disorders. So the second paper that was published in Nature, I'm sorry, this was in Lancet, is identification of risk loci with shared effects in 5 major psychiatric disorders, as I said, autism, bipolar, major depression, schizophrenia, and ADHD. And they have about 30,000 cases and controls. And the main findings are, besides that all of these disorders are very heritable, they found that there were co-heritabilities. In families, they found that some of the subjects of schizophrenia had also in the same family or the same genes were present in subjects of bipolar disorders. And they have some co-heritabilities in major depression, schizophrenia and autism is the last, and not so much schizophrenia and ADHD. So that's something to think about. And then I'm gonna come back to this point later just to say, it's a problem in terms of diagnosis because it could cross over to other disorders, okay? So we'll come back to this. But also I want to tell you, bipolar disorder is also another highly heritable disorder. And the same group, the bipolar group, found 30 loci. And the one that you can see here, or not see, the one that you can see is this calcium channel. It's also present in bipolar. But you can tell here in chromosome 1, we don't have MIR137. And in chromosome 6, we don't have the immune system. So they are common, but also separate genes and genetics that can distinguish the two of them. There was a more recent study in 2008 that showed that if you look at schizophrenia and bipolar patients versus controls, what you find is that there are some, like I said, some variants here. So the bipolars and in this graph, they are in blue, and the schizophrenia subjects are in red. And anything that is higher means it's more, if this variant contributes more to schizophrenia, and anything that is in blue, this contributes more to bipolar disorder. And I give you an example of the top changes, and 3 of them are listed here. This is the SNP name. It's a number of the SNP. and 3 of them contribute to schizophrenia, and these 2 here, the middle 2, contribute to bipolar disorder. But what is interesting is you have more schizophrenia risk in the patients with bipolar. You have more likelihood of have bipolar with psychosis. You can have bipolar 1 with psychosis or not psychosis. So it's something informative to think about. Again, the similarities between these disorders, but without psychosis, they are separate disorders. So how did this information help us, or did this information help us? On other way, what is the efficacy of this polygenic risk score? So all the variants together, all the risk alleles that were identified by the GWAS study, what do they tell us about diagnosis, okay? And this is what it tells us. It tells us something, but not as much as we need. So if we have all these common risk alleles for schizophrenia, they can tell us out of 20% of the risk. For the 1%, now we are 20%, they can explain when they have all together. If they have disease, think about all the SNPs, these are deciles, but the decile 10 is 100% of all thousands of SNPs. So and this is various, this is the green is the population from US and Australia, the other is from Sweden and Denmark. So it tells different things in different populations, but it doesn't have the specificity to predict who will have the disorder. So genetics does not explain it all. And we're gonna see how we can improve that. The other thing, two recent studies published last year and this year use the same schizophrenia SNP, risk SNP, thousands, and they apply to patients for healthcare systems in the US. So these patients were admitted to the hospital and they volunteered, they gave the DNA so they can do all this SNP analysis. And the good news is you can see, well, schizophrenia, the majority, again, if you look at the highest level here means the strongest finding was schizophrenia, bipolar disorder, depression, and other psychotic disorders. But we also have anxiety and mood disorders that we haven't talked about. And we also have substance addiction, tobacco, major depression, suicidal ideation. So there are other things that the patients that have some of these SNPs came to the hospital for. So it's not ready for, for just someone comes to the hospital, quick test, and you know what this patient has. So it's not there. But again, hold on to the end. I'll tell you how we can improve this. So, okay, so I've been telling you about the missing factors contributing to schizophrenia, and I'm going to tell you about epigenetics. Epigenetics, let me move here, is an emerging field of science that studies heritable diseases caused by the activation of genes. So the genetics here in the DNA, here's the chromosome and the DNA, each chromosome is a piece of DNA. So if you unwind the piece of DNA, you can see all the bases like I explained before, the TA, GC, and AT. But in addition to, they are the compaction of the chromosomes. Here is called a structure, very compacted, that's called nucleosome, or it's more open, the chromatin is open. And when you have open chromatin, is transcriptional active. When it's compacted, it's not. And the regulation of this, of how the genes are expressed or not, has to do with these epigenetics. And epigenetics are histone modifications around in the nucleosome, and DNA methylation around the DNA methylation in cytosines. But it's also important to know, and I think is that there are medications that we didn't know, but have been tested, and we know they work via changes in methylation. And they are lithium, valproic acid, lamotrigine, olanzapine, quetiapine. So these are medications that can help regulate the expression of the genes. And I'll give you one example next, because we studied something similar. And I forgot to tell you the title and the reference of this paper that was published in March of this year in Biological Psychiatry. Is Genetic Modification in Schizophrenia-Related Disorders. And I think the title is very appropriate. Molecular scars or environmental exposures and source of phenotypic variability. And I'll tell you what they are. What are the insults? Why these we think are scars? But first, let me give you a gene that is in gut. One gene is an enzyme that synthesizes GABA, and GABA is the main inhibitory neurotransmitter in the brain. Glutamate is the excitatory, and this is GABA puts the brake. Okay, it's important because it puts the brake. And GABA, this gene is under DNA methylation. So if you see all these pentagons in here, means it's methylated. And what this and other authors found is that during maternal stress, maternal immune activation because of flu and other infections, and low postpartum maternal care, plus the genetic variants, can increase the DNA methylation in this gene, which ends up having less GABA. And we know that in schizophrenia, we have less GABA because we studied this in 2008 in a paper in the American Journal of Psychiatry, showing that all the GABA genes, GABA, this gene and other genes related to GABA function were decreased. So it's something that is important to, so we know this is one of the mechanisms of schizophrenia is neurotransmitter alterations. But what we know now is that valproic acid, which is called Depakote, it comes in different names, can do this at least in animal models and cell lines. So there could be an opportunity to use a mood stabilizer and anticonvulsant drug like valproic acid to kind of restore the balance of GABA to glutamate in the brain. And so now what I want to tell you is now we have the epigenetics and the genetics, and now we can think as schizophrenia not just genetics, but there are epigenetic factors, and schizophrenia, starting in the conception, and it's after adolescence is when you see the disease. And in another study, this is a nice review, you can see that you start, they call sometimes the first hit is genetic, and the second hit could be early environmental insults like prenatal infections, or the later in adolescent, if you have high cannabinoid use, especially in adolescence, it's not recommended at all. And then you have other effects. So you need multiple hits in order to have the disease. So it's not, we don't know exactly how many you need. And again, this is something I presented, all these contributive factors. So here a family history, the genetics, and all of these, Rubiola, influenza, everything that is above 2 is significant. Bereavement, CNS damage, obstetric complications, preeclampsia, all of these could increase the risk. And I said could increase the risk because it's not certain. None of these is certain. We are talking about predisposition, okay? It's a number game. Nothing is 100%, even if you have all of this, okay? So I want to have this clear. And for those that are interested, today I learned, the Brain and Behavior Foundation funded my research as an assistant professor, a young investigator award, and then as a associate professor and independent investigator award, and they are a fabulous resource for all our research efforts. And they have groundbreaking channel of the lesson psychiatry and lecture by John Luby that is going to be registration is free. If you want to attend, let me know. It starts after October 30 and is run by the Brain and Behavioral Foundation. All right, so now I'm going to wrap up with the most interesting part, that I think this is the future. We are not there yet, but I see there's hope. And the hope is kind of how you combine neuroimaging and genetic approaches. And this has to do with the work that I have done, always in collaboration with people at MRN and now in GSU, Georgia State University. And I want to tell you that a former postdoc in the lab of Vince Calhoun, now an associate professor in GSU, she was the leading expert in combining neuroimage and genetics to understand the structure and function, brain dysconnectivity in schizophrenia. And the way you can see this, you cannot tell which, if I tell you which brain is from the patient with schizophrenia, I wouldn't know. They look the same, and they are, by most part, the same. So you have to be able to apply several techniques to understand this brain, not only structured, but functional. And with Jean, what we did is we started working at combining functional MRI and SNP data to look at using a method called parallel ICA, the independent components analysis, and which she has continued to use. And also now she's doing genetics, epigenetics, transcriptomics, and brain-based phenotypes. And this is a review that was published a couple of years ago. So I think this, as I said, if you combine behavior and genes, you can understand better behavior, neuroimaging and genes, you can understand this better and you can think about better treatments. This is another, my colleague Jessica Turner and Vince Calhoun also have these studies in a part of the Enigma consortium, another consortium looking at structural neuroanatomy. So this is a structural MRI. And what they found that the frontal cortex in here, frontal cortex to an insula, sorry, the insula is right here, it has to be inside here, are associated with a component in chromosome 12, and they are, next week, a new grad student is going to be presenting. Next week is the World Congress on Psychiatric Genetics, and October 20th we have a poster, virtual poster, and we are going to present a follow-up of this work. But I think it again shows how you can combine genetics and neuroimaging. And I want to leave you with this one, which is the work of many, many years of Vince Calhoun that has done a lot of work looking at multimodal neuroimaging. And multimodal means fMRI, functional MRI, structural MRI, and this is diffusion MRI for white matter. So this is for function, this is for structure. MEG is again for neuronal connectivity. EEG is from the top of the skull. Oh, sorry. And the idea, if you have all this data plus genetics from all these patients, obviously it's not easy to have it in the clinic because this is a very extensive study. It's only at the research phase, but you can get data-driven using algorithms developed by Vince, Dr. Calhoun. You can get subtypes and you can segregate the disorders. And in here you can see the wearables segregate just with all of this information, all of these, healthy controls in one cluster, bipolar disorder, schizophrenia, schizoaffective with depression, and schizoaffective with mania. And here is a way of depicted all these different groups. So I think, again, this is the future is not available for diagnosis, but I think the more we can use some of the information and making a step forward for the clinic, it will be helpful, at least, and this is my hope for the future. So I'm going to stop now. I don't know, it's probably time to entertain questions, but before that, I want to thank Carrie Wright, that she's now at Johns Hopkins University, all my colleagues, Vince Calhoun, Jessica Tonin, Jinjo Liu, Jiaju Chen, and Dr. Juan Bosillo, who helped us with the patients, to recruit the patients for these studies. NIMH, NARSAD, which was the first part of the Brain Behavior Foundation. And then the COBRE grant that we have been having this COBRE grant of schizophrenia for the past 15 years. So, and I want to say this is in the memory of Silvia Beatriz Perrone, who is my sister, or was my sister, and my mom. I owe it to them because my sister had a very severe accident as a child, very young child, and then later developed schizophrenia. And my mom that took care of her till her death in '84 when I came to this country. And I think in all the moms, I want to say, we do all what we can, and I'm sure that all of them are doing the best. In the 50s, it's it's the moms, it's the environment, it's a combination of factors, and many of them are not our faults. We cannot say which person will have it. What we can do is prevent. Maybe cannabis use would be a good idea if they are adolescents, and then hope that these medications, like I said, that can change epigenetics, maybe will be available as well. So with this, I want to entertain questions. So I'm going to stop sharing unless you need me to go to specific slides, but I want to stop here. Thank you.
Thank you, Dr. Bizzozero. Excellent presentation. Do we have any chat questions? Or I see we don't, but do we have any questions from Facebook, Jessica?
I'm not seeing any so far.
Do we have any questions from our Zoom audience before I ask my questions? Cameron.
Tom, thanks. Thank you, Doctor, for your presentation. I have really two questions about epigenetics. And the first is, I think I read on one of your slides that there are heritable changes. Does that mean that an epigenetic change in, say, my generation can be passed down to my children's epigenetics? That's the first question. And then the second is, it sounds like epigenetics can be changed, not only in the womb, but also later in life. And I'm curious if there's any way to characterize, I guess the significance. Is it more significant, these changes in the womb, for instance, or can later in life epigenetics be changed, so drastically that a disease becomes possible where it wasn't before? So, thank you.
Thank you. Excellent question. So I'll start with the first one. And the first one, this is very new emerging field. And the reason I said it can be passed on, and this is something that is very, very recent and very new, is that epigenetics changes, there are epigenetic changes in the semen that can be passed to the offspring. And especially, if there are, I know for nicotine use that can be passed, alcohol use can be passed. With schizophrenia, we don't have as much information of how they can happen, but I know for sure there are several conditions they affect, the semen, the information in the semen that can be passed to the offspring, mainly in animal models, but it's a possibility. Then the second question was how soon you need to change, if you could change epigenetic changes. Well, I would say if we knew, the problem in this case, we have the first insult in utero, probably, or right after. And then you don't know if the boy or girl will have schizophrenia till they are after 15, 16, 17, 18, 20. So it's hard to predict, though I have to tell you, there are ways now, and I didn't say this, I should have added that, but I have collaborated with others looking microRNAs in blood, and actually have a paper in the alcohol journal showing that there are distinct microRNAs in the maternal serum, of moms that drink heavily alcohol. This is another of the disease study, fetal alcohol disorder, and that correlate with maternal drinking. So there are ways to look at epigenetic modifications that are not in the brain, but in blood, and that's more accessible. And there's studies done in my department, Department of Neurosciences has the New Mexico Alcohol Research Center, and they are looking also at epigenetic modifications, and we are working on that too. So the more we know what happens in blood, is better, because right after the child is born, you could take, blood and understand, what happens with microRNAs or other changes, DNA methylation. There are all possibilities. There are, again, non-diagnostics today, but I think as more people and more researchers start publishing on that and giving us more indication that these could be predictors, those changes could be changed, could be added. Obviously, we have to know when it's safe to give a drug like valproic acid. And again, I said this is, I'm sorry to say this is like 10 years from now, probably we'll have that information, maybe 5 years, who knows? I'm trying to say I'm optimistic, but we need much more research, enabled to do that. And maybe someone else knows they are doing now, and I'm looking forward, what happens, the presentation from the Brain and Behavior Foundation to see if they have ways to address this. But it's very, very significant question, and I'm sorry I don't have all the answers right now, but I'm hopeful.
Thank you, Doctor. Do we have any other people with questions? And you can use Zoom to indicate you want to ask a question, but I don't see any others. So I do have a few questions, and
okay.
I wanted to ask them. I was very intrigued by the use of fMRI and genetics together. How long do you think it might be before that becomes a more standard diagnostic tool when someone presents with symptoms of schizophrenia?
Okay, so the problem with all the research studies, in order to go mainstream, they have to be a lot of publications. So, and to see the value. As I said, schizophrenia is diagnosed by the clinical symptoms. So if a psychiatrist diagnoses schizophrenia, I'm sure there are really good tools. They have a lot of the Diagnostic Statistical Manual, they have a lot of DSM-5, they have all these categories. So they have really the chronicity, how many symptoms, how long. So it's kind of very precise. But I would say, so the problem is diagnostic schizophrenia in the prodromal phase before, and that's when you have someone at risk, diagnose whether this person, or tell you whether this person is a trajectory that will end in a schizophrenia or bipolar disorder before it happens. And that's gonna be critical, because in that case, you don't have the clinical symptoms yet, but it's gonna happen that, so if you could predict, use fMRI, structural MRI, genetics, epigenetics, and those individuals of high risk, but that they have not had the clinical, full-blown clinical symptoms, then that could be really helpful, because then you can start medication earlier. And that would be really the advantage of that. fMRI, you can do it in a clinical, fMRI would be easy to do in a clinical setting, because you can do a structural MRI and fMRI in a clinical setting. The clinical setting does not have the expertise to do fMRI and don't have the recommendation. Is not a standard of care. Is not a standard of care for diagnosis. So it has to be more and more studies need to show that this could be useful, and then it will happen. So we are just at the research phase. We are only in that level in which you recruit select group of patients, and you can do that. There are also child psychiatrists here. Rochelle Lenroot is an outstanding child psychiatry and researcher that's here at MRN, and she's studied population of trauma and populations that have risk, trauma, all type of trauma also have some increase the risk for schizophrenia and other diseases later in life. So, we are having in New Mexico, we have really good scientists, and our collaborations in Georgia State are really good too. So maybe you can ask them in the future to elaborate more on those. And if I can facilitate those, I'll be happy to do that.
Thank you very much. And one more question. I was very intrigued about epigenetics, which can change throughout somebody's life. And one of the things we heard from other speakers is, and this was, I think, Dr. Frank Pieri from UNM, that when someone gets diagnosed with schizophrenia, they start out with, they get on one group of drugs that work, but then over time, those drugs may not work anymore, and they go to a different, hopefully they find another treatment regime that works with the drugs. And I just wondered, is it possible that one of the reasons why drugs stop working is because there are epigenetic changes that change the underlying nature of the illness, and so a different set of drugs is necessary, that in the course of the disease, could the epigenetics be changing?
Well, that's another very important question, Tom. Yes. What we don't know is that every time a patient has episodes of psychosis, usually they could get worse. Also, the medications have side effects. Mm-hmm. All the medications you have heard, Parkinsonism, you can hear even the, other medications that are more the new medications that are not the typical, like Haldol and the typical, they always have side effects. And yes, the body can change every time you give any drug. The body tries to change and kind of compensate for the drug. So that would be something that it could happen. Whether it's epigenetic, I don't know, because it hasn't been studied why you need to change medications. But the hope would be that using genetics and epigenetics in the future, they can understand what type of medication, maybe like I said before, personal medicine, what this type of medication may be important, may be the best for this type of patient with these genetic and epigenetic changes. So there are, as I say, there are personalized medicine for other disorders like cancer. So it's not out of the realm that there's going to be personalized medicine for mental illnesses.
Thank you. We got one more question that came up from the chat, and I'll read it to you.
Okay.
I have read that the absence of choline may be responsible or contribute to schizophrenia. Is that something you can comment on?
Okay, so the absence of choline, and okay, so the absence of choline, you mean in sorry, the person is in neonatal or in adult?
I would say both.
Okay, all right, so choline is essential. We need acetylcholine it's an important neurotransmitter for brain development. It's also important for thinking, thought process. And you know that acetylcholine binds to nicotine receptors. And those receptors, that's why patients will self-medicate by smoking, because the nicotine acts into those receptors. So if there are deficits in choline, and I know the case in fetal alcohol disorder, that the moms had deficits in choline, many times because poor diet and other drugs. So it's a complicated, bad nutrition, drugs during pregnancy. So they have the ability to give choline to the babies after they are born, soon after. And this is not a standard of care, but I know there are clinical trials, and the beauty of giving choline is that the blood-brain barrier that protects our brains is not developed at the very young child, the baby. So that could be. It's hard to know if the patient is deficient. It will be, again, choline, lecithin. I mean, choline, we get it from greens. All the lecithin is any food that has greens, we can have choline. Phosphatidylcholine is something that is important for choline. We can degrade phosphatidylcholine, which is lecithin, which is in the greens, and you can get choline. Then in the adults, it's hard to get choline in the brain, as I said, because the blood-brain barrier will prevent choline to go inside because it's a charged molecule. But in the babies could be. But first you have to establish whether the mom was deficient or the person is deficient in choline. So I don't know about I can check it out and find out in PubMed if there are studies on that in schizophrenia. I'm not aware of them, but I can get back to Jessica or to you, Tom, to get the answer to the person that asked the question.
Thank you very much. I'd like to thank our speaker, if we could. I realize applause doesn't work real well on Zoom, but nonetheless, I very much appreciate your coming and speaking with us. This is absolutely fascinating. And I think one of the things I was most fascinated by was just the extremely high quality of the work being done at UNM. And I think that's partially reflected by the fact that your students, postdocs, and collaborators are moving out to other great institutions and being able to work from there as well. So there's nothing, I think, more satisfying than seeing the people that you help learn move on and do well. And you seem to have been very successful at that. So congratulations in that regard.
Thank you. Yeah, mentorship we value mentorship, and I'm very proud of all the trainees that I trained through the years. So I'm very happy for that. They are doing really well.